Hey everybody, it's Zach from Myshire Farm, and we are back again with another video to help you on your journey with Coturnix quail and becoming more self-sufficient. In this video, I'm actually not going to be talking. It's going to be Christy from the Duke University. That's right, she did another genetics video for us. It is fantastic. She's going to be talking about inheritance, egg size, egg shape, egg color. She's going to be talking about body weight and kind of breaking a bunch of things down, what you can get with different things and how that kind of works as far as the inheritance behind it all. It's really, really good. And then I kind of wanted to simplify that just a little bit more for people that don't really know a lot about genetics. So at the end of this video, I'm going to come back and I'm going to give you a couple things that we do here that might help you with your jumbo breeding program. So I hope that it helps. I hope that you enjoy it, and we'll see you in just a little bit. Thanks.
Hi again, everybody. I'm Christy. For those of you who didn't see my first video, I've done another video on Zach's channel about some more general genetic topics. And today we're going to talk a little bit about inheritance and how that might impact the health of your quail or the size of them, the shape and colors of their eggs, and all that fun stuff. So let's get started. So today we're going to be talking about inheritance. And inheritance is basically something that we refer to when something is being passed down from a parent to an offspring. So it is only one direction, parent passing down to offspring. And in our case, this is DNA from parent being passed down to the DNA of the offspring. And so just as a little refresher from the last time, a parent will have 2 copies of every single gene in their genome, in their total DNA. They'll have 2 copies of everything. So 1 copy, 2 copies for each parent. And your child will get one of these and combine that with the DNA that they inherit from their other parent, also 50%, and this will produce this unique offspring. This, very simple mechanism creates a lot of diversity in offspring. So, your children don't look identical to you, and that is by design. So essentially predicting this based on parent DNA. Typically, they do one of 2 things. They can either be in a relationship where one is dominant and one is recessive. So we can have the dominant recessive relationship, which essentially, if you have a dominant trait, it will win. It will beat out the recessive trait. So one appears mostly independent from the other. The alternative to that is dosage, where basically you can have a certain amount of something and your 2 copies can simultaneously produce that thing and therefore produce a lot of it. You can hit a certain point where you'll have a certain trait because you produced a lot of it. You can produce some of it, and maybe one of your traits is working and the other one isn't, where you'll produce something intermediate, and you can produce none of it, and you'll produce the opposite end of the spectrum of producing a lot of it, whatever that may be for that given trait. This will be based on usually thresholds. Thresholds determine appearance. And in case I say this word without, catching myself, there is this word alleles. And an allele is essentially the feature that you're inheriting from a parent. So you have 2 alleles for every given trait because you are inheriting 1 allele from mom and 1 allele from dad. And so this is basically our trait. So in case I use that word without catching myself, I want you guys to know what I mean. Allele essentially is interchangeable with the word trait. Now let's get into, how you might predict the DNA of the offspring from the DNA of the parent, like I said earlier. Essentially, what you can do is this little toy example, which comes from a man named Gregor Mendel, who did a lot of research on inheritance well before we knew what DNA was. And so essentially, he did all of his work based purely on observation, which is a really important part of science. This man, Gregor Mendel, was a German monk, and he was growing pea plants. And when he was growing these pea plants, he noticed that the pea pods, which we'll draw like this, I'm not a particularly gifted artist, tended to be green or yellow. And so you have this pea pod that's showing some different, traits, different phenotypes, different features, just as far as you could tell, somewhat randomly. But he wasn't really happy with the idea that it was random, which we'll get into in a minute. But the other thing that he was noticing was that these pea plants had flowers, which again, not a gifted artist, which could be red, or they could be pink, or, they could be white. And he wasn't sure what was causing these things. And so he tried to understand how breeding one feature against another feature might produce a predictable pattern. And so what happened if you were to pollinate a plant that had green pea pods with a plant that had also green pea pods? Or what happened if you crossed a green pea pod plant with a yellow pea pod plant? And what happened if you crossed a red flower plant with a white flower plant or a pink flower plant? What would happen in the offspring? And essentially came up with something that we still use today because it is a great example of the simplest versions of inheritance. And so he used what we Punnett squares, which some of you might have a flashback right now. I apologize in advance for any biological or scientific trauma involved by seeing this little square, where essentially mom has trait X and trait X, dad has trait Y and trait Y. And, you can kind of predict what the offspring might have just based on pure statistics. So we're going to go through some of these examples because it's kind of fun.
So let's say that a green pea pod plant, that you're wanting to cross with another green pea pod plant. So we got green pod plant. We'll call green pod 1 mom. And mom, just so happens that her 2 traits, her 2 alleles, are 2 green alleles. So 2 traits that say be green pea pods. And the way that we refer to this in these examples is by coming up with a single letter system or at most a double letter system. If you don't mind, so too messy in this little tiny example. So in this case, I'll say that a big G means green trait, and mom has 2 of them. And so in these examples, we're talking about dominant recessive relationships. Just for the green and yellow examples, we have 2 dominant or 2 greens in this in mom. And let's say for dad, who also has green pods, that he actually has a big G and a little g. And so he has 1 dominant and 1 recessive. But he is still green. So he has 1 green and 1 yellow in this example. So now we have the basic traits of mom and dad. But what might their offspring look like
if you were to cross-pollinate mom and dad? So let's do a little example. So we have our little square that we're going to draw. So mom will go up here, big G, and dad will go here, big G, little g. And so essentially what will happen is we will predict it by just doing a simple cross. So we'll say this offspring potentially has 2 big G's, so does this offspring, but these 2 offspring actually have a chance of being big G, little g. And that's because dad, half the time, will give them a little g instead of a big G. So 50% of the time, or 50% of the offspring statistically, need to be at least big G, little g. So we'll have 1 dominant and 1 recessive allele or trait. But another 50% will actually be big G, just like mom. So 50% will look like dad and 50% will look like mom. And in this example, they will all still be green. So 100% of offspring will be green, like both mom and dad. But what happens if I cross offspring of these? And so this is now, because we're using plants, inbreeding is actually really informative for plant genetics. And so let's say that Gregor Mendel, in his curiosity, decided to say, okay, well, let's cross one of these, offspring, and he just so happened to pick one with big G, little g, with another one of these offspring, which just so happens to also be big G, little g. So now we're crossing 2 offspring with big G, little g. And in this scenario, we have 1 out of 4 that will become big G, another one that will be big G, little g, a second one that will be big G, little g, and then the last one will end up with little g, little g. So in this scenario, we have 50% that end up being big G, little g, and green, 25% that end up being big G, and green, and another 25% that end up being little g, and these guys are our first yellow plants of this experiment that we're doing. These are our first yellow pea pod plants from this experiment that we're doing, where we now have some not green plants coming from a cross, 2 crosses, in fact, that only had green plants involved. And so now this is why I say that inbreeding plants like this can be really interesting, because you end up finding these recessive traits. And actually, this is the exact same reason why it causes health problems in humans or animals if you inbreed repetitively, because traits like these can sometimes be harmful. Instead of just coding for yellow, that DNA sequence might code for something harmful. And the same thing goes for quail as well. After a couple of generations, they start to get just less hardy if you're continuously inbreeding them, which is why it's good to have, outside populations brought in so that you can keep everybody healthy. Now, remember the other example. So remember that was the dominant recessive relationship, but we had this other example that I call dosage. So what happens when you're working with dosage alleles or dosage traits? So essentially what I mean when I say dosage is that if you have a low concentration or a low dosage of whatever that trait produces, whatever the DNA of that trait produces, you will have, let's say, trait A or appearance A. Let's say you have medium dosage. You can have trait B. and high dosage could have trait C. So this can be, in various examples, changed in a bunch of different ways. Sometimes after you hit medium dosage, you just have the same trait for medium and high. Sometimes it's the opposite, where unless you have very high dosage, you will have the same trait. And so medium and low in this example would be the same trait, where high would have a different trait. But in general, this is the one that's most fun to look at, because this is how you might end up with something like what we were looking at a minute ago, where we were talking about red, pink, and white flowers. And typically, these dosage traits are because of chemical reactions that happen to produce whatever the trait is. So in this case, a chemical reaction that might produce red pigment in the petals of the flower, if it doesn't have enough things to work with, might only produce pink because it's producing red, but only a little bit of red, and might produce white, but only because it's not producing any color at all. And so white is really translucent. It just doesn't produce pigmented color in the petals, and therefore the flower appears white. So what we can look at now is what would happen if we did a little Punnett square, which is what those crosses are called. I don't know if I mentioned that before. Those are called Punnett squares. We can do a little Punnett square for, red and pink and white flowers. Let's say we have a red flower. Which means that both of its copies will be big R. And so producing a whole lot of that pigment. So both of those copies will be big R, and then we have a white flower, where both of those copies are little r. And so let's do our little Punnett square for these. So we have big R, big R. We have little r, little r. And so for all of these, we're going to have big R, little r combinations. Yeah. And so 100% of our flowers, so we have big R means produce pigment, and little r means produce no pigment. And so for every single one of these, we're producing some pigment. And if you need a lot of pigment to be red, so then 100% are producing only some pigment. So 100% are going to be pink flowers in this case. But let's say we cross two pink flowers. So now we have pink flower is going to be R, R, big R, little r. And let's say that we do two big R, little r's for this Punnett square. Big R, little r, big R, little r. Now we have big R, big R, big R, little r, big R, little r again, and then little r, little r. And so now what we're going to have is 25% that are red with big R, 50% that are pink with big R, little r, and 25% again that are white with little r, little r. And so now you have this like wonderful mix of colors happening when you started with all pink flowers. And so this is what Gregor Mendel kind of pioneered, was that there is a method to the madness of why you can cross a red flower with a white flower, somehow get pink flowers. And then if you cross pink flowers against pink flowers, you somehow get red, white, and pink flowers. You can see how that would be baffling. So Gregor Mendel did a lot of work to just understand the fundamentals of inheritance without knowing anything about genetics or DNA outright, because as far as they literally did not know that DNA existed at that point. So this was incredible work and very detailed work, and we have great records of it. So now you might wonder how this is in any way relevant to quail breeding or quail farming. Well, I saved the difficult examples for last because let's say we're talking about quail body size. Talking about quail body size, the problem is that body size is not necessarily a very predictable trait. It doesn't behave like these might, or even like the dominant recessive might, because it doesn't just have one location in the DNA controlling it. So body size is what we call a complex trait, which essentially just means that there are many places in the DNA that contribute to body size in every way. And so you will have parts of your DNA that will be coding for things that make something small, and parts of your DNA that will be coding for making things big. And they all just kind of work each other out as, you know, whatever it is that you're talking about. In this case, quail is growing and developing. They work each other out in their system. And so body size is very difficult to predict for that very reason, is that there are thousands of places in the DNA that are actually controlling for that specific trait. Because when you think about it, you're not just controlling the physical size and shape of the animal, you're also controlling their organ size, their bone length, their head, their, spatial patterning for, where all of the parts of its body are. And so it's a really complex thing to discuss. But the good thing is that you don't necessarily need to know all of that to get a big animal. So I have a Great Dane, and it just so happens that Great Danes are very, very big dogs. But at the same time, you could breed them with a Chihuahua, and what you'd likely see is something in the middle. And so while it's not necessarily straightforward inheritance, it's not the most reliable version, it is somewhat predictable. So, this is how, Zach has this jumbo breed of quail. They are jumbo because if you breed them consistently against each other, you're going to get large animals. And, you will reliably get large animals that continue to be large, no matter who you're breeding them with, so long as it's another, somewhat large animal. The other thing that is interesting about quail is egg size and pattern. So the really cool thing about patterns in all of biology and all of genetics is that they are very fun to understand. So there are some patterns, like for example, a butterfly wing. So let's just draw a little butterfly wing where you might have, just finish the butterfly, where you might have like a little dot here, and then this like nice little pretty pattern that occurs around the dot, and then another dot here, and this pretty pattern again. And for the same kind of butterfly, that might be fairly consistent. And a different species of butterfly will have nothing even similar. And typically what happens to make that is your DNA can control not just what it's telling you to do, but where it's telling you to do it. And so the DNA over here is saying produce B, and the DNA over here is saying produce A. And because these cells get caught in the middle, they produce both, and it creates this unique combination. And so this is, behind a lot of patterning is the overlapping of different instructions. We have, instructions that are telling it to do one thing over here and instructions that are telling it to do another thing here. And the conflicting instructions produce something else entirely. You have this like uniqueness produced by the combination of A plus B. And that is a really fun topic in biology, and it's called patterning. The reason I bring this up is because if you've seen a quail egg, you can have very unique patterns. You have, for example, there's one of the quail that I have right now is producing this really cool like galaxy effect, which kind of like leaves this like little line here. And the top part has this like very faded brown background. The bottom part has a white background, and there's speckles all over the place. But it leaves this little remnant line there. And that is just consistently her egg pattern. That's what that looks like for her egg. I've also had a few of the birds that I've gotten from Zach produce green eggs or greenish blue eggs. The speckles might be different between egg to egg, but in general, the appearance looks somewhat identifiable and consistent. And so there's not really a lot of understanding at what that is, attributed to, but it is a really cool feature of eggs is that they have these very unique patterns and combinations. They're still consistent to the actual bird. So I think that the takeaway here is that this is a really complex system. But if you're breeding, the female that produces these eggs, her daughter is more likely to have this kind of pattern than the daughter of some other female. So for example, that female that I told you about that had the blue-green eggs, one of her daughters actually produced exclusively blue-green eggs. And so that's just kind of a complex trait that we don't have a great understanding of. In general, inheritance is a really cool thing to study. It's really complex a lot of the time, but then there's these nice, like simple, neat examples where you have a threshold or a dosage trait, and sometimes you'll have this nice dominant recessive trait, and they're like really cool to look at. It was wonderful to see you all again. I am going to let Zach take over from here and let me know if you have any questions. Me and Zach will talk after this video is posted, and yeah, if you guys want to see more, just let me know.
Okay, so we're back. I hope you enjoyed it as much as I did. Hopefully you'll have to watch it 2 or 3 more times like I did as well, because I'm just a little slow at this stuff. But she did break it down really, really well, and I actually understand a lot more going forward as far as what George and Jenna tell me what to do. But I did want to break a couple of things down as far as our breeding program goes. So at the end of the video, she was talking about how A is saying, okay, be just like this, look just like this, and B is saying, be just like this, look like this, and that little sliver in between is what the offspring will actually do. I think that's very important for a lot of people. So if you're not really into a lot of genetics, if you're not into biology that much, or you don't really understand it, that's okay. I want to completely simplify it down to looking at them and judging them aggressively is very, very important. But the whole aggressive calling thing, I don't really agree with. The reason I'm saying that is because I think that the majority, not all, but the majority of quail always have a purpose. So for example, and I'm just going to give you one, and if you want to know more about this, please comment below and I'll do a separate video, and maybe Christy can do some as well as far as the breeding program goes. But for example, let's say we weigh out our jumbos. Well, we have a standard, right? So let's say you're weighing your jumbos and you're saying, I don't want anything in my cage under 12 ounces. Okay, got it. So you weigh them and you weigh 100 of them. And so you've got 10 that weigh between 10 and 12 ounces, right? And then you've got 4 that weigh under 10 ounces. Okay, well, that 10 to 12 ounces could be a separate cage that you're trying to increase the size of something else. That's a great starter for you. So you've got all your jumbos, you've got your standard, and then those 10 that are between 10 and 12 ounces, well, they don't hit your standard for the jumbos, but you can easily put those with something else and start breeding that next color a little bit larger. And then the ones underneath that could easily be a feather sexable mixed cage. or breeding it back to something, or whatever the case may be. So the point I wanted you to take away from this is looking at them, weighing them, checking them out. You know, if you don't like the egg size or the egg shape or the egg pattern or something, you really don't want to use it for that specific purpose. But that doesn't mean that it can't go towards a different purpose. Now, obviously, they can always go to butcher, they can always go to falconry food, they can always go to reptile food, sure, absolutely. But if you're trying to get your numbers up, being aggressive and being very determined on what you want into that specific cage is very important, absolutely. But that doesn't mean it can't go towards another cage just for egg production or eating eggs or whatever the case may be. So I think a lot of people lose sight of that and they forget about that. But if you have any questions and you want to know more about it, I don't want to take any more of your time, and I appreciate everybody watching this long. But if you have any other questions or want to know more, please comment below and we'll make sure we do more videos or answer your questions in the comments. So thank you very, very much to Christy from Duke University. I am so happy and proud that she decided to do this for us. So thank you very much. I thought it was fantastic. Make sure you show her love and comment below how much you appreciated the video. And hopefully I didn't confuse you more. But until next time, everybody, as always,